Great, thank you. All right. So I wanted to start with just a quick overview of what Epstein-Barr virus is. It is a human herpes virus, so similar to the varicella-zoster virus that causes chickenpox, for example. And herpes viruses are really good at living in our bodies forever. So Epstein-Barr virus, where it kind of hangs out in our body after we're infected with it, are in one of the immune system cells, in the B cells specifically. You can see that in the top figure here. I'm trapped by wires, or I would kind of point a little better. But so in any case, it becomes what we call a latent infection. Once you're infected with EBV, you're always infected with EBV. The body's not able to clear it, it just controls it. And during one's life, the EBV can become reactivated for various reasons that can happen. And then the virus will begin to shed, it's active in the body, you can pass it to somebody else, and the immune system tries to get it back under control. So EBV is really interesting because it's more of a recently discovered virus. It was first discovered in 1964, and it was discovered in cells from Burkitt's lymphoma, which is a kind of cancer. And it was shown in 1968 to cause infectious mononucleosis, which I show down here at the bottom, some of the common symptoms of infectious mononucleosis. Now, most people are infected with EBV when they're young. So when you're an infant or a toddler, preschooler, when you're going to daycare, kindergarten, in those ages. But there are some people who escape EBV infection early in life. And when the EBV infection happens early in life, it's asymptomatic for most people, or for a young child, there might be like a fever or something that doesn't look abnormal in terms of like, kids get sick all the time. So no one would kind of think like, oh, this child has just been infected with EBV. But for people who are not infected with EBV as a child, about half of the infections will occur later in life. So as an adolescent or a young adult. And typically that will happen when someone leaves home to go to college. So now they're around a whole bunch of new people, or to join the military, again, new people. And people who haven't been infected are at high risk for becoming EBV infected at that point. And in about half of those infections that occur at that stage of life, about 50% of them will manifest or will present with infectious mononucleosis. And I'm sure everyone in here maybe has known somebody who's had infectious mono, if not even yourself. So it had been noted that the epidemiology of EBV and the epidemiology of MS was pretty similar. And one aspect of it that was similar is that the infectious mononucleosis that was happening, people were infected and having infectious mononucleosis about maybe 10 years or so before they may onset with symptoms of MS. So a number of groups, studies worldwide have looked at the association between specifically having had infectious mononucleosis and MS risk. And what this figure is what we call it's a meta-analysis figure. So each line here represents a different study. And you can see that most of the, or all of the dots, the red, black dots that go down are on the right side of that line, meaning that infectious mononucleosis, or having had a history of infectious mononucleosis, is associated with an increased risk in MS. And in this case, it's a twofold increased risk in MS is what we see across these studies. And I know that this comes from a paper from 2010, but there have been more studies that have been done recently that find the same thing. In fact, in MS epidemiology, I would say the infectious mononucleosis association is the most consistent, reliable. I mean, we see it in every population we look for it in. We see it no matter how, the infectious mononucleosis is recorded. Is it if people self-report it, or if it's actually laboratory documented that this person had infectious mono. It's almost like no matter what way we slice this twofold increased risk seems to fall out. So it's a very consistent very consistent association. So an interesting question is, from a causal perspective, if EBV causes MS, then we need to see that someone's infected with EBV and they get MS. If someone is not infected with EBV, they don't get MS. So in this chart here, again, this is a bunch of several different studies. This is another way to combine studies. But in these studies, what they did is they looked at people with MS, assessed whether or not they were seropositive for EBV, and compared them to a set of controls. And you can see in the red boxes at the bottom, when they did this, they found that less than 1% of the MS cases were actually EBV negative, as compared to 6% of the of the controls were EBV negative. And the 6% is about what we expect to see in the general population. It's around 94, 95% of all adults have been infected with EBV at some point. And so this translates into an extremely low risk of developing MS. I have an odds ratio on the side, which basically says that your odds of developing MS if you are EBV negative is virtually zero. So these were some of the first sort of indications that EBV may be causally related to MS, but at this point, it's still just an association, and we couldn't say anything more than that. So for the past 20 years, what we've really been wondering is, does EBV infection occur prior to the onset of MS? And do EBV negative individuals have a near zero risk of MS? And this has been challenging to study for a number of reasons. One is that we can't conduct a randomized control trial. We can't take a group of people and randomize half of them to be infected with EBV and half of them not to be, and then follow them for the next 20, 30, 40 years and find out what happens. We also currently don't have a vaccine against EBV, so we can't even do a vaccine trial to see if that would prevent MS at the moment. So what we really needed is a large EBV negative population that we could follow over time. But because EBV is so prevalent in adult populations, it's very difficult to identify that group of people. Because of the high seroprevalence, MS being a relatively rare disease, we really needed a large cohort of young people to study this in. And so we did that in our US military cohort. So the US military cohort is active duty military. Every 2 years or so, once they enroll, they provide a blood sample for a variety of reasons, for HIV testing, for worldwide deployment situations, and the blood is then sent to the Department of Defense Serum Repository, which since the early 90s has been collecting samples. They get about 2 million samples a year, and they have over 60 million samples on over 11, 12 million people at this point. So it's a really large group and a really great group to find an EBV negative cohort, because those who haven't been infected by the time they've enrolled the military, will show up negative, and then we can see whether they develop MS, or not develop MS, excuse me, whether they become infected with EBV, and then who goes on to develop MS. So it's almost like a naturalized trial in a way. So what we really wanted to determine was whether EBV infection precedes MS in time. So in order for EBV to cause MS, it has to occur before MS. So we were interested in looking at the clinical onset of MS. So did EBV infection happen prior to any symptom onset? And then did the infection also precede an elevation in a molecule called neurofilament light, or NFL? And neurofilament light is part of the skeleton of nerve cells. And so when nerve cells are damaged, the neurofilament light is leaked into the spinal fluid and then also leaked into the blood. So we're able to measure those levels, and those will be indicative of damage to the central nervous system. And we were also interested in looking at just, immune dysregulation in the preclinical phase, because one of the criticisms of the association with EBV is that, well, maybe people who are going to develop MS or who have MS just have this blatant sort of out of control immune system. Maybe they have reactions to all kinds of viruses, and maybe this EBV is just an artifact. So we wanted to take a look at that and see if we could tease that out as well. So as I mentioned, we conducted this study in active duty US military. It's a very large cohort of young adults. And they have repeated serum samples available from the repository, because over the course of their career, they do contribute a sample, like on average, once every 2 years or so. MS is a medically disqualifying illness in the military, meaning if you are diagnosed with MS, you are referred for a medical separation. And at that point, we are able to get a hold of medical records of individuals who are being separated from the military due to MS, and we're able to review them, gather some information about when they onset, when they were diagnosed, what their MRIs look like, what other tests and things may support the diagnosis. So as I mentioned, the repository is quite large, 62 million samples, over 10 million individuals, and it has taken us 20 years to document MS in 955 active duty personnel. So we have the 955 cases, and we have to select controls for them. And this is just showing that we've matched them on sex, age, race, ethnicity, dates of blood collection, and branch of military service. Meaning that if we had an MS case who is female, her control is also female. If she's 29, her case is 20, or her control is 29. So they're exactly the same on all of these matching factors. So in this group of people, we had 35 people who went on to develop MS and 107 of the controls who were EBV negative in their very first sample collected after they enter the military. So we have 130 or 142 people who are EBV negative. That's our EBV negative cohort. And so what we did is we took that cohort, took all of their serum samples, and we measured EBV in them. So in their first sample, they were all EBV negative, and then we followed them to see if they became infected with EBV, because their antibody levels to EBV would change if they became infected. So the median age at first sample collection was 18 years old, and the median age at MS onset in this group is 28. By and large, the vast majority are relapsing remitting MS, and given the nature of the US military, most of them are men. Though interestingly, when we look at it, we still see the 2-to-1 female to male ratio in the military, but the absolute number is greater just because of the makeup of the population. So in the second slide, or in the second figure, C, you can see what we've plotted here is the serum sample. So the first one, the earliest one that was collected, then the next one, and then the third one, which is the closest to MS onset. And you can see that that first sample was collected, on average, for most people, about 10 years prior to their symptom onset of MS. So in that first sample, collected 10 years prior to, is EBV negative. So you can see that in here in figure A, the first serum sample here, so we see seropositive sample percent up the side, and then which serum sample it is along the bottom. And the first serum sample, everyone was EBV negative. Then we follow them, so the cases, the people who go on to develop MS and the people who do not. And essentially what this is showing is that there is a much higher rate of primary EBV infection in individuals who go on to develop MS than those who do not. So nearly 100% of the people who developed MS seroconverted or became infected with EBV sometime after that first sample but before before the symptom onset of their illness. And in the controls, there was like it was around 57% seroconverted around that same time. We also looked at other herpes viruses just to kind of see, like, is this specific for EBV, or is this happening with other viruses as well? And another virus that's very similar in how it moves in the population is cytomegalovirus, which is CMV in the second. And you can see that among those who were negative for CMV, the rates of infection over the same period of time were very similar. It was really the EBV separation that stood out. So then these are relative risks. And so using the information on the previous slide, we're able to calculate what the relative risk is of seroconverting for EBV and MS risk. And so you can see here that compared to the people who stayed EBV negative, we did have controls that didn't become infected at all. And we had one case who did not show any sign of EBV infection. So compared to that small group of EBV negative people, those who did become infected with EBV over the time period had a 32-fold increased risk of developing MS as compared to those who remained negative. And then in the CMV, you can see similarly, it actually is interesting in going in the opposite direction. So the CMV negative group, it looks like there may actually be an inverse association. So if you're infected with CMV, it may actually provide some protection or lower your risk slightly of MS. And this has been shown, it's been picked up in other studies as well, and we don't know what it means. We don't know why that's happening, but it is an area of interest.
So
yes.
You said there's that 32 times what you said, that greater chance of getting MS.
Yes.
Wow. Is that, have you ever seen anything else with that big?
Not for MS, but it is, you know, similar for like smoking and lung cancer.
Got it.
Yeah. And I actually think the smoking and lung cancer relative risk is a little bit lower. But when you're talking about these numbers, you know, the size, it probably doesn't matter too much, but yeah. What about Parkinson's? So as far as I know, Parkinson's is not thought to have a viral cause. And I don't think EBV. You
thought it was similar.
Similar. Oh, you know, the epidemiology of Parkinson's is really interesting because when you compare to MS, because it does have, like, it's thought of more as a disease of older age. And so Parkinson's tends to onset more in 60s, 70s, actually 70s, 80s. It's much more rare to have Parkinson's begin at such a young age. And when it does begin at a young age, it usually has a really strong genetic component to it. But I'm not aware of any studies linking any viruses to Parkinson's disease.
H1N1 in 1918, there was an outbreak of Parkinson's in the mid-20s, which they referred to as a post-encephalitis type of thing.
Right, right. Yeah, no, that, yeah, I'm
unclear the data.
Right, yeah, I have heard about that actually, yeah. Yes.
Oh.
I'm sorry, can you say it again?
My mother's father had Parkinson's disease. I always wondered because everything could have been handed there.
Right, right. Yeah, you know, I know, because genetically speaking, you know, MS, you know, falls more into the autoimmune category where Parkinson's does not. So you tend to see like autoimmune diseases, maybe, you know, other diseases like rheumatoid arthritis or lupus or type 1 diabetes. I'm not aware that there's so much overlap between Parkinson's and MS. I mean, there could be something, but it could be, you know, a rare situation, or it just could be coincidence. Yeah. Okay, yes.
let's say a high school or college student gets infectious mononucleosis, you know, then there's a trigger for MS to potentially come, I think it's 6% or something like that. What can be done between the onset of the infectious mononucleosis and the onset of MS to minimize or eliminate the MS? Is it targeted vitamin therapy, any type of diet, or anything that you can use to minimize the risk?
That's a great question. That's
there's no
there's
vaccine, and that's the eventual goal. But
right.
For what we have to deal with now, what can be done?
Right. So I want to make a distinction between a relative risk and an absolute risk first. So the relative risk, we're comparing this now to like people who are EBV negative. So it's relative to their risk. The general risk, when you think about just like the general population, they have about 1 in 200 lifetime risk of developing MS. So it's taking that 1 in 200 and multiplying it by 32 if that primary infection happens in adolescence. Not necessarily, well, one thing importantly that we don't know here is we don't know at the time of primary infection whether this individual experienced infectious mono or if it was an asymptomatic infection. So we can't tease that out here, unfortunately. So, because it would be really interesting to know if infectious mono specifically at this point, really increases the risk of MS even more than this. But what I would say is that we know that vitamin D, for example, is associated with a decreased risk of MS, as is not being obese in early life, and of course not smoking. Also, cigarette smoking is associated with an increased risk of MS as well. So I think if you want to target, if somebody there has infectious mono, if they don't already, making sure that their vitamin D levels are within a high normal range. So either they're taking a supplement or there's somebody who practices, safe sun exposure, maybe out in the sun for 10, 15 minutes before they put on sunscreen in the summer. Making sure that, if it's a person who is obese or has weight issues, that maybe we're helping them trying to, get some of that weight off. And if they're a smoker, certainly talking to them about stopping smoking. And if they're not, certainly they don't want to pick it up. But in terms of specific like targets or nutrients or medicines or anything to reduce that risk, we're just not there yet. Yes.
How this relates to the higher risk, the higher latitude people are further north, they have more MS.
So we're talking about if I heard you correctly, the latitude gradient. So there's more MS in the north than in the south in general. Yeah, so it's really interesting because historically, EBV infection has also shown that north to south gradient. And one of the thing people think maybe behind that is just historically, generally, the more hygienic north, the less hygienic south. More like there's more opportunity for people to have been exposed to EBV by this gradient. So, and the early studies have shown that people coming from the Northeast into the military or into college have less EBV infection than people who are going into those same places from the South. But I will say that has changed. I think we're sort of converging on very similar infection patterns in both parts of the North and the South. So I think it comes down to some other factors now, and we actually have seen an attenuation of the latitude gradient, where it's not as strong as it was in those early military studies that they did, looking at where people were coming into the military from. So it's almost, in the United States anyway, it looks like it's pretty much disappeared, that we're on equal footing. But certainly that was actually one of the observations that led to the hypothesis that EBV may be associated with MS, because they have that similar geographic distribution. Yes.
I'm from Aberdeen, Scotland, the northeast, and there's a high percentage rate of persons up in the top of Scotland who have MS for some reason. And nobody seems to know.
Yeah. Yeah, it's, right, because there are some places where, small-ish geographic locations do show a really strong gradient too. And, maybe it has something to do with EBV. There could be different strains of EBV. There could also be, we know diet, actually, with the vitamin D and also with, we're actually conducting, we've published on this and they're doing a little bit more research on what are called polyunsaturated fatty acids, which are, like you may have heard of as omega-3 or the fish oil fats, or the good fats. And, we're seeing that people who have a higher intake of those types of fats seem to have a lower risk of MS. So it's, possible maybe there was something in the, I don't know what the dietary patterns are in Scotland specifically, but maybe the southern Scottish area is more fish, or they're more likely to take fish. I mean, I have no idea, but there are those kinds of little subtleties that could also be contributing to the within country.
Fish and cheese.
More cheese.
We love cheese.
Yeah. Are there any, yes?
Is your group conducting any clinical trials that progressive patients in this room might be eligible for?
So our group does not conduct clinical trials. Well, so because we're not in a clinic, we are not connected with a clinic. So, like places like MGH or Brigham and Women's and those kinds of places that do clinical trials, they have, the patient population to work with. We, in MS, don't do any clinical trials. It's all observational data, meaning that we have very little interaction with people with MS, but we have data from them and are able to conduct these kinds of studies.
So you mentioned omega-3.
Yeah.
Would you, any sense that helps people who have MS?
So just between you and me, it's not
Yeah.
published yet.
Fair enough. I'll take
This
it.
is like Vegas.
Right. No, but it was presented at a conference, so I think I can, yeah. So we do have a study. We actually work with one of the pharmaceutical companies, and we have access to blood samples and data that were taken from the patients who were part of that study. And we actually measured levels of fats in their blood, and we have outcomes. So these were people with a clinically isolated syndrome, so they've only had one attack that could potentially be attributed to MS. And so we were looking to see, do they develop MS? The other outcomes were like, I think relapse rate, or what their score is on their EDSS. And so we see that a nice association with blood levels of alpha-linolenic acid, which is a plant-based omega-3, seem to have a decreased rate of relapse over the 5 years that we're looking at, and also less likely to convert to clinically definite MS. So it may be that early in the disease having that, and interestingly, we also see it with two of the fish fats as well. The DHA and EPA, the two from the fatty fish, also seem to be showing similar association. So hopefully that will be actually like published in the next year or so. But yeah, so at least in early MS, we need to look also at people who were in some of these trials who came in already with like relapsing remitting. Yeah. Yes.
I was diagnosed at 21, and I remember back in high school, I had mono. I was really sick, and I got a black box on top of my brain. And my old neurologist said, yeah, you must have a closed low back. So, it could be like infection.
He said you once had a what?
Mono.
Oh, mono. Okay.
Back in high school.
Yeah.
So
I was really sick.
I don't know if this is legitimate or appropriate because it's a health question, but it might be interesting to see a show of hands, people in here who have MS, how many had mono?
That's if you want to share that information.
I would raise my hand.
I did.
Mhm. Yeah. They were quite, yeah, a few. What's that?
I had a stroke at 33.
Mm.
My grandmother had a stroke at 33.
Mhm. Yeah.
That don't mean nothing with you.
Yeah.
Thank you.
I'm sorry.
That's it. Thank you, Anthony.
I'm having a hard time. Yeah. Sorry. I just, yeah.
Epstein-Barr.
How many people have it?
Yeah.
Yeah, so in some populations, it's 100%. In like the United States, it's more like 94, 95%. So it's a ubiquitous virus that, you know, I would guess that if I had to place a bet, I would bet that all of us in this room have it, or at least most of us. Yeah. So, and that answer. Are we? Did you have a follow-up? No. Okay. Shall I keep going? Okay. No, that was great. No, I really, I appreciate the discussion because it's, you know, I'm always, you know, giving these talks to other scientists and other MS specialists and things like that. And I really love the opportunity to talk with the actual patients and the people who are experiencing this, and answering any questions that I can. So I like the informalness of it, I guess is. So someone had asked, I think earlier, like, have you seen associations this strong? And I said, no, we haven't. And one of the things that always comes up too is that, well, could this be explained by something else? Is there, something else that causes MS, but also is responsible for EBV infection somehow, maybe some genetic factor or something along those lines? And I will say that next to EBV, the strongest risk factor we have is the genetic risk factor for MS, which is about a 3-fold increased risk of MS. And we don't need to go through the details of how we calculated these numbers, but basically what this chart is saying is that for something to explain completely the EBV association that we see, meaning that it's not really EBV, it's something we haven't discovered yet, it would have to have a 64-fold increased risk or association with both MS and independently with EBV infection. And so far, no one has come up with any plausible explanation or alternatives that would explain the association completely away. So this also strengthens the argument for causality here, that we don't have another explanation for this. If you think if something has a 64-fold increased risk with MS, we would have found it quite easily by now. So we don't feel it confounding or something else can explain these results. I'm sorry. EBT? Epstein-Barr virus.
Okay.
I'm sorry. Right. Okay. So again, some of the criticisms we've had on our previous EBV research has been, well, you know, does MS just increase the risk of being infected with EBV? Which is, you know, one of the possible explanations when we look at blood of people with MS, and they do have evidence of EBV infection. We don't know when that infection happened. Did it happen before the MS, after the MS? And then there's been the argument also, is there this general just, you know, immune response to many different viruses, and you're just picking up one of them, but it doesn't actually mean anything specifically causal for MS. So in this study, we looked at this in a couple of ways. Did you have a question? Was there
The military always checks for one.
Does the military check for EBV? No. No. It is, you know, they may measure it after somebody finds themselves in the, you know, medical unit for infectious mono, but it's not screened for as part of a condition for entering the military. So the two ways that we sort of tried to go about assessing whether we really had something that was occurring prior to MS occurring. And one was, again, to measure these levels of this neurofilament light chain. Like I mentioned earlier, that these are molecules in the nerve cell. They're sort of like the part of the skeleton of the cell. So when the cell is damaged, it releases some of these skeletal molecules, and we're able to pick them up in the blood. And we also had this opportunity to measure basically an immune response. So measure antibody levels against pretty much like all the viruses that infect humans. So about 200 viruses that are capable of infecting humans. We were able to sort of like screen their blood to see what viruses they're having a reaction to that's different than the controls. Okay, so in terms of perceiving the neurofilament light increase, which would indicate that there's something going on at the, you know, at the level of the central nervous system. There is something happening to the nerves in the brain and or spinal cord. And when we measured the neurofilament light in the same blood samples that we had measured EBV in, on the left side, you can see these are the results for the people who went on to develop MS. And on the right side, the people who did not. So they were all EBV negative in that first sample. So the one that's sitting on the 0% line. There, and these are actually, sorry, these are like changes in NFL, right? So we start with the first sample. So the first sample is what it is. So there's no change. It just is a value. And then we measure it again at the second sample, which may be around the time of EBV infection. Well, is around the time of EBV infection. And you can see that there's still no change. It's what this means is that that second dot means that the NFL in that blood sample was nearly identical to the one in the first sample. There is no change there. So in their last sample that we collected, and we know they've been EBV infected, this is some time after EBV infection has established itself, you can see that in the MS cases, there is like a 60% increase in their NFL levels from that first sample where they're EBV negative. So basically what that's telling us is that when they're EBV negative, their NFL level is at 1, whatever it may be, probably usually pretty low, because it's low in people without neurological disease, as you can see in the controls. And then we don't know exactly when they were infected with EBV. That's one thing. We only have 2 samples, one negative, one positive. It happened somewhere in there. So that's why we say around the time of EBV infection. So with that sample, there was still no change in the NFL. And it wasn't until after they had been infected with EBV for some number of years that we were able to detect that increase in NFL. And now importantly, just to remind you, all of these samples were collected prior to what we determined as their time of symptom onset. So at this point, presumably, there's no, these individuals are not experiencing yet the symptoms of MS, because the samples were collected, the study was designed to take samples that were collected prior to that time point. And you can see in the controls who did not develop MS, their NFL levels stayed the same across the same time period. So they were infected with EBV, but there was no indication of neurological damage. Is that?
So you could take somebody who got mono and at a certain point regularly measure their NFL and maybe see, oh, it's spiking, something's going on.
So that's a great question. And it gets to whether or not we can use NFL as a potential biomarker for assessing someone's risk for developing MS. The research is not there, certainly yet to use it in a clinical way. A lot of work is being done on NFL because I think it would be amazing if we had something like that. But it is, yeah, it's a great question, and it's a question that a lot of researchers have as well. Yes.
You're saying it's that symptom everyone doesn't know when something begins.
Yeah. So okay. So I can clarify how we do that. So we review the medical records. We get the same set of records that the military does, that they use to evaluate whether this person should be discharged due to an MS diagnosis. And I gotcha. I agree with you. There are records where we're scratching our head and we're like, did this when, could this thing that they report from high school, could that have been it? We're constantly trying to evaluate that. But there are some people and some records where it seems, at least the way it's reported, very clear cut. Like they were fine up until this moment when they woke up and this happened, and they can't, they have no other symptoms. We try to be very conservative. I want to say, because we know how important it is to have blood collected before symptom onset. So we do, when we're looking at these records, we will let a case go. If we know it's an MS case, but we can't figure out from the records when their symptoms reasonably could have started, they're not included in our study. And similarly, if anything existed prior to entry, so if somebody did report something similar, like say, oh, they had this exact same thing happen to them when they were in high school, we can kind of go, all right, so maybe this existed really before they entered the military, but it just wasn't picked up. And so we let those cases go as well. We try very hard to ensure that those samples, and is it perfect? No, absolutely not. But yeah, but that's, I feel like it's the best that we can do with the information that we have. Yeah.
You said it's not positive.
I'm sorry?
You are saying that's just not the cause of it.
Just not the cause of it.
What's not the?
Having mono is not the cause of your MS.
Right. Well, so we think that the EBV infection is somehow causally related to MS. For some people, if they've had mono as a result of that infection, that seems to increase their risk more.
No, because one of in high school,
Mhm.
when the service right after. And after they got out of the service, they come up with the event.
Mhm.
Nothing to do with military.
So when that, I don't know about specific military exposures. I mean, that is another area of interest, but the person that you're describing, I think, is someone who, right, had mono before they entered the military, did not have MS while in the military, right, and left the military and then developed MS? No.
Yeah.
Yeah,
They're not tracking.
exactly right.
They tried to sue them.
We can't find those people. Because those people, once they leave active duty military, they go into the veteran system. And once they're in the veterans, one thing I'm learning about the military, well, actually the government in general, is that no organization likes to talk to another organization, even within this larger military thing. So we actually can't easily, if at all, link the information from the Veterans Administration to what their active duty data are. So for the veterans, we have no data, so we cannot. So later onset MS, we can't track it. We have no way, unfortunately. We're trying. We're trying with ALS, right? Which is an even more rare disease. So it's you know, it's the government. There's lots of bureaucracy, and it's no one's talking to each other. So, but yeah.
Maybe at this point, because it is getting close to noon,
Okay.
people can hold their questions till the end, and Dr. Munger can finish her presentation, and then if we have questions,
Okay, perfect.
That would be great.
Yeah, perfect. And I'm happy. I have all day, so I'm happy to stay and chat. Whatever you know. So I have a feeling. Okay. All right. So that was so we felt that this particular set of analyses has showed us that, okay, so the EBV infection is occurring prior to any evidence of a neurological pathology going on. And so then we were interested in, okay, so is this just associated, what we're seeing with EBV, is this just associated with a more general immune dysregulation? Are there other viruses for which there is a difference in response between those who went on to develop MS and those who did not? And we can see here, so we had hypothesized based on all the previous work that we might possibly see something with EBV, but we didn't know what else we might see something with. So here you can see, so the two red outlined boxes go together and the two blue ones in terms of comparison. So in the top are the pre-onset samples that were taken from the MS cases. And at the end there, you can see that's the antibody response to Epstein-Barr virus. And the same response in the controls is circled in the bottom panel. So people with MS in their pre-onset samples and preclinical onset samples had a much stronger response to EBV than the controls did. The blue box was actually samples that we had that were collected after the onset of MS, and you can see that the same difference persists in those. Can you see? I don't know why. I'm so sorry. Okay.
We'll make sure everyone gets the slides.
Okay, that would be great. And I'm happy to, you guys can find me by email too. I'm available. Oh, okay. I just was worried that maybe the people who are sitting over here all of a sudden can't see the slides. But okay. So in any case, it's very specific, you can see, for EBV, because if you look at the other, even though they're not like this blue here and here, they're showing a very similar pattern between the cases and controls. So the people who went on to develop MS didn't have some sort of aberrant immune response to these other viruses in the same way that we were able to pick up EBV. So my summary and conclusions, what we've taken away from this study is that, primary infection with EBV, so the first time you're infected during young adulthood, greatly increases MS risk. And EBV negative individuals are strongly protected against developing MS. And it can't be explained by what I had mentioned was confounding. So it can't be explained by any other factors, or what we call reverse causation. So meaning that we showed that in a linear way with respect to time, that the EBV infection is occurring before there's any indication of MS occurring. And there's also no evidence for generalized immune dysfunction response to viruses in general. It's very specific for EBV. And so our group has concluded that that our evidence outside of having a vaccine or being able to conduct a trial does provide the strongest evidence to date that MS is a rare complication of EBV infection. And I think, so I mentioned the vaccine, there are trials that are going on, not for MS specifically, but for, can they prevent mono or prevent EBV infection at certain stages in life, and what benefit might that have? And then, of course, the role of EBV in driving MS activity and progression is also a topic that's top of everyone's mind right now, trying to figure out, okay, so if EBV causes it, is it sort of like a hit and run, or is it still there and leading to other outcomes for MS patients? And quickly, just to acknowledge my group at the Harvard Chan School, the first picture is Dr. Alberto Ascurio, who is our fearless leader. And then two of our other research scientists, Mariana Cortez and Shetel Jornovic, who've done a lot of work on this particular analysis. And of course, we have many military collaborators, medical school, and it was actually the University of Basel in Switzerland that did the NFL levels. So with that, like I said, I'm happy to take questions, happy to stay as long as you want me to stay. Thank you for listening. Thank you for the great questions so far.
Thank you.